This video will present topic 6.4, Translation. Following the production of a primary transcript, or pre-mRNA, in the nucleus, and that primary transcript's modification in RNA processing, the second stage of the expression of a gene will use the information encoded in the mature mRNA to assemble a specific sequence of amino acids. Outside the nucleus, in the cell's cytosol, a variety of components act upon the messenger RNA to produce a polypeptide. Complete polypeptide is then modified and conformed into a specific three-dimensional shape, promoting the polypeptide into a functional protein. Before we move into the details of translation, let's briefly review the types of RNA that play major roles in gene expression. Although there are dozens of types of RNA that have been discovered, there are four that we need to be reminded of. mRNA, or messenger RNA, produced in the nucleus, carries genetic information from there to a ribosome in the cytosol. Transfer RNA, or tRNA, are free-floating in the cell's cytosol and shuttle amino acids from there to the ribosome. Their structure includes a three-nucleotide sequence, an anticodon, that is complementary to a sequence of mRNA nucleotides, a codon. Ribosomes contain RNA and are complex structures found free-floating in the cytosol or attached to the endoplasmic reticulum. They are responsible for providing a location at which mRNA and tRNA can temporarily base pair and amino acids get joined together. These three types of RNA are directly involved with the process of translation, while small nuclear RNA, which is found in the nucleus, has already served its purpose in removing the introns from the primary transcript, leaving only the exons in the fully mature mRNA. Therefore, we won't see this type of RNA playing any role in translation. The genetic code, the information carried within an organism's nucleotide sequence, and the manner in which it's acted upon, is a fantastic example of the evolutionary interconnectedness of all life on Earth. The fact that a given codon specifies the same amino acid in plants, fungi, animals, bacteria, and protists is exceptionally strong and important evidence linking all life on Earth to a common ancestor. The genetic code is a key part of the history of life, and the set of rules used by living cells to translate information encoded within genetic material evolved as early as the origin of life, well over 3 billion years ago. In biology, when mechanisms or systems or processes are said to be conserved, it identifies ones with early origins that have changed very little over evolutionary time. Gene expression is one such process. Fireflies carry a gene allowing them to produce an enzyme called luciferase, resulting in a phenomenon called bioluminescence, and they glow. Since the manner in which fireflies express that gene is identical to the way in which tobacco plants express genes, they can be genetically modified to carry and express that enzyme. Green fluorescent protein is produced by a species of jellyfish, resulting in them glowing when exposed to ultraviolet light. The mouse on the right has been genetically modified to carry that same gene, isolated from the jellyfish. About 10 years ago, researchers at the University of Wyoming came up with a way to incorporate the genes that spiders use for producing silk into the genome of goats. With an interest in studying the potential applications of spider silk due to its strength and elasticity, a number of medical uses, such as making artificial ligaments and tendons to repair injuries, and industrial applications like improved car airbags and even bulletproof vests, may be possible thanks to the near universality of the genetic code. Ribosomes play a central role in translation. Some found floating in the cytosol, and others attached to the endoplasmic reticulum, will bind to the messenger RNA to begin translation. But before we go into the details of that process, let's take a closer look at the structure of a ribosome. A single ribosome is actually comprised of two subunits, one being larger than the other. They are not so cleverly called the large subunit and small subunit. The large subunit has three functional sites. These are locations at which specific events of translation will occur. The first of these three sites is called the A site. Officially named the amino-acyl tRNA binding site, this is the location at which transfer RNA molecules will enter the ribosome, bringing their amino acids with them. The peptidyl tRNA binding site, or P site, is where peptide bonds are formed between amino acids, linking them together. During translation, this is also the site at which the growing polypeptide would be located. The final site of the large ribosomal subunit lacks a chemical name and is simply called the exit site, or E site. From here, tRNA molecules that have donated their amino acid to the polypeptide under construction would then exit the ribosome. With an understanding of the structure of a ribosome, let's now see it in action. The translation part of gene expression has three distinct stages. The initiation stage is what brings together all of the components involved with polypeptide assembly. The two subunits of the ribosome, the messenger RNA, and the collection of transfer RNA molecules, one at a time, bringing their respective amino acids to the ribosome. Beginning the process is a complex of the small ribosomal subunit and the first tRNA, which, thanks to its anticodon, is complementary to the mRNA's start codon. The 5' cap that was added during RNA processing in the nucleus is what indicates directionality to the mRNA and therefore acts as an attachment site for the small subunit. Beginning at the 5' end, the small subunit slides down the mRNA until the start codon, AUG, is encountered. This sets what is referred to as the reading frame. From this point forward, the mRNA is utilized by the ribosome one codon, or 3 nucleotides, at a time. To complete the initiation stage, the large ribosomal subunit binds to the small subunit using energy from the hydrolysis of GTP in the process. It is critically important to make note of the positioning of the large subunit. You should notice that the first tRNA is located at the P site of the ribosome. Now, here's an animation from the textbook publisher to show us what initiation of translation looks like.
The message in mRNA is translated into a protein in the cytoplasm. First, a transfer RNA, tRNA, arrives, carrying a specific amino acid. The small subunit of a ribosome attaches to the mRNA. Now, the large subunit of the ribosome attaches.
Following initiation is elongation. This stage happens to be the longest since it is involved with joining together the hundreds, thousands, or even tens of thousands of amino acids to form a polypeptide. Here's what happens. The currently unoccupied A site receives the next tRNA made possible by codon-anticodon matching. Here's the codon chart for reference. The codon CCG would specify the amino acid proline. UAU codes for tyrosine, and GGC would call for glycine. The connection between amino acids, a peptide bond, is formed between them using GTP energy as the ribosome pulls the mRNA down by exactly one codon. This results in the growing polypeptide being positioned at the P site of the large subunit. Also, after the codon shift, the A site is unoccupied and the tRNA with no amino acid, also called an uncharged amino acid, is ejected from the E site. Let's continue with the earlier animation to see what this elongation looks like.
A second tRNA docks, bringing another amino acid. The ribosome helps to form a covalent bond between the two amino acids. The mRNA shifts, and the first tRNA leaves. A new tRNA brings another amino acid. The ribosome helps to form a new bond, and the process is repeated. Notice that one end of a tRNA molecule has a set of 3 bases called an anticodon that pairs with complementary bases on the mRNA. The other end of the tRNA carries a specific amino acid. Different types of tRNAs carry different amino acids. In this way, the message in mRNA is translated into a specific sequence of amino acids.
Elongation continues, with the ribosome drawing the mRNA through itself, one codon at a time, until one of the 3 stop codons is pulled into the A site. Transfer RNA molecules do not exist for any of the stop codons. By extension, that would also mean there is no corresponding anticodon and clearly no amino acid for a stop codon. So what does happen at a stop codon? A protein called a release factor binds to the stop codon and releases the completed polypeptide, the mRNA, and the 2 ribosomal subunits. The completed polypeptide will now undergo a collection of modifications to conform it into a functional protein. The polypeptide is a protein's primary structure. In order to assume secondary and tertiary structure, modifications are made to it. Some of these changes are occurring concurrently with translation as the polypeptide is coming out of the ribosome. The most common changes to a polypeptide include the addition of functional groups. This diagram exemplifies a few of those, such as a phosphate, hydroxyl, methyl, or acetyl group. Carbohydrate and lipid additions can also be made. The takeaway from this is that these changes alter the chemical and physical properties of different regions of the polypeptide so that it can bend, fold, and shape into a specific functional conformation. Some functional proteins are complexes of multipolypeptides. Collagen is a quaternary protein that is important structurally, providing strength and elasticity to connective tissues. It is comprised of 3 interwoven polypeptides, twisted around each other. The well-known hemoglobin is a quaternary protein that is made of 4 polypeptides, 2 alpha chains and 2 beta chains. Finally, many proteins are further modified by the addition of cofactors. A cofactor is a non-protein chemical compound or metallic ion that is required for an enzyme's role as a catalyst. They can be considered helper molecules that assist in biochemical transformations. Cofactors can be divided into 2 types: inorganic ions, such as the iron found in hemoglobin, and complex organic molecules called coenzymes. Coenzymes are mostly derived from vitamins, such as the B vitamins, and other essential nutrients in small amounts. Some of a eukaryotic cell's ribosomes are attached to the endoplasmic reticulum, while others are floating freely in the cytosol. The manner in which these 2 types of ribosomes carry out the process of translation is identical. What differentiates them then is the ultimate destination of the protein being manufactured. If a protein is to be secreted from the cell or will not be a component of the cytosol, translation will occur on a ribosome attached to the endoplasmic reticulum. These so-called bound ribosomes assemble the amino acids into a polypeptide that will be further processed by the Golgi apparatus. The completed protein would then be packaged into a vesicle or lysosome. In contrast, free ribosomes, those floating in the cytosol, translate the information in an mRNA for proteins that will remain in the cell. These proteins will also undergo post-translational modification before becoming active cytosolic proteins or being incorporated into some other organelle. A lot of our understanding of the processes involved in the manufacture of proteins comes from our study of bacteria, especially E. coli. Much of what is involved in building a protein occurs in identical ways in prokaryotic and eukaryotic cells. In both cell types, information from DNA is copied into an RNA molecule by RNA polymerase, which is then acted upon by a ribosome to assemble amino acids in a specific order. However, there are two quite significant ways in which protein synthesis in prokaryotes differs from that in eukaryotes. The first difference is that transcription and translation are said to be coupled. This means that those two processes would occur simultaneously for a given gene. As RNA polymerase is transcribing a gene and an RNA molecule is being assembled, ribosomes are able to attach almost immediately to the free end of the still under construction RNA. This is possible due to the fact that there is no nuclear membrane separating the site of transcription and the site of translation, as there would be in a eukaryotic cell. The second major difference is that RNA processing does not exist in prokaryotic cells. Neither the 3' poly A tail nor the 5' cap are necessary in bacteria. After all, there is no nucleus for the RNA to exit, and only one end of the RNA molecule, 5', is suspended in the cytosol to which the ribosome could attach. The lack of RNA processing also means that the structure of prokaryotic genes lacks introns. The sequence of DNA nucleotides is transcribed directly into a functionally mature messenger RNA. Prokaryotic DNA is exon only. Up to now, we have studied the central dogma and its steps, whereby DNA is used to produce RNA, and that RNA is used to produce, eventually, a functional protein. However, back in 1980, a new class of virus was discovered that operates contrary to the central dogma: retroviruses. Retroviruses carry RNA as their genetic material. But this isn't what makes these viruses interesting, since there are many different kinds of RNA viruses, including the cold viruses, influenza, Ebola, measles, and the coronaviruses. The uniqueness and importance of a retrovirus can be attributed to an enzyme it possesses, called reverse transcriptase. Based on the name of that enzyme, you could probably imagine how it functions. It uses its RNA as a template to manufacture DNA, in other words, the reverse of transcription. The single-stranded DNA assembled by reverse transcriptase acts as a template to produce its complementary half, yielding double-stranded DNA. The viral DNA is then incorporated into the host cell's genome within the nucleus. Now that viral DNA is part of the host cell, the benefit to the virus is twofold. One, were the host cell to divide, additional cells with viral DNA would be produced. Second, when the virus's genetic information is expressed, more virus are produced, allowing the infection to spread to new cells. We've come to the end of topic 6.4. Thanks, and take care.